In the previous video, we discussed glycolysis and how it aids ATP production in the body. We established the fact that when one molecule of glucose enters the glycolytic pathway, there is production of net ATP of two molecules of NADH, and 2 molecules of pyruvate. Now we established the fact that the ATP produced from glycolysis are used by the body for energy production. The NADH which is produced from glycolysis also enters the outer mitochondrial membrane and joins the electron transportation system in the inner mitochondrial membrane for ATP production. Now the question is, where does the pyruvate find itself, or where does the pyruvate go? Now the pyruvate, which is the main product of glycolysis, have several fates in the body. 1. In conditions of no oxygen or low oxygen concentration, the pyruvate can be channeled to the production of lactate. This pathway mostly occurs in a vigorously contracting muscles with an enzyme lactate dehydrogenase. This condition is an anaerobic respiration because it involves no oxygen. Now, some microorganisms possess an enzyme pyruvate decarboxylase and alcohol dehydrogenase. These two enzymes work together in a two-step reaction to convert pyruvate to ethanol in condition of no oxygen or low oxygen concentration. Also, the pyruvate can be converted to some amino acids by transamination. Example, it can be converted to alanine by the alanine cycle, whereby you add an amino acid or amino group to the pyruvate to form alanine, like an amino acid. In the conditions of an oxygen availability or in an aerobic condition, the pyruvate enters a cycle we call the Krebs cycle. The Krebs cycle is the main cycle of production of several molecules of the electron equivalent, which is the NADH and the FADH2. These electron equivalents are what is used in the electron transport chain for production of net ATP. Several of them. Mass production of ATP occurs by oxidative phosphorylation, which occurs in the inner mitochondrial membrane, and the electron equivalent mostly come from the Krebs cycle or the TCA cycle. The pyruvate is first converted to acetyl-CoA by an enzyme we call pyruvate dehydrogenase complex. This enzyme or this complex ensures the conversion of the pyruvate molecule to the acetyl-CoA. Now, the acetyl-CoA can come from several routes also in the body. This can come from beta oxidation of fatty acids, or also deamination of some amino acids like, let's say, glycine or even cysteine. Now let's see how the acetyl-CoA produced from pyruvate through the glycolysis or from any of the discussed routes enters the Krebs cycle. Now, when the acetyl-CoA finds itself in the Krebs cycle, the first reaction is more of an aldol condensation reaction by reacting itself with compound we call an oxaloacetate. This one is a 4-carbon compound and acetyl-CoA is a 2-carbon compound. The two undergoes an aldol condensation reaction with the help of the citrate synthase to produce a citrate. The citrate is a 6-carbon compound with 3 carboxylic acid functional groups. That explains why the cycle is called a tricarboxylic acid cycle, because it has an intermediate compound with 3 carboxylic acid functional group. The next reaction is from the citrate. There's an isomerization of the citrate to an isocitrate through enolization. So an aconitase enzyme tends to lose water from the citrate and convert it to cis-aconitase. The aconitase also undergoes hydrolysis to produce net isocitrate. Now, for the first site of ATP production or the NADH production occurs in the conversion of isocitrate to alpha-ketoglutarate. So the isocitrate produced here is first converted or oxidized to alpha-ketoglutarate by an enzyme isocitrate dehydrogenase. This reaction is more of an oxidation reaction because NAD+ is used to oxidize isocitrate to alpha-ketoglutarate, followed by a decarboxylation reaction which is led by loss of carbon dioxide. So the first site of NADH production is conversion of isocitrate to alpha ketoglutarate. Now the alpha ketoglutarate also undergo further oxidation and decarboxylation to produce succinyl-CoA. Here the NAD+, which is an oxidizing agent, gets reduced to NADH, followed by decarboxylation reaction, which is catalyzed by alpha ketoglutarate dehydrogenase complex. Now from succinyl-CoA, there's production of an equivalent of ATP, which we call the GTP, guanosine triphosphate. Also has potential energy production potency like an ATP. So upon conversion of succinyl-CoA to succinate by an enzyme succinyl-CoA synthetase, GTP is produced. This reaction is more of a hydrolysis reaction that leads to the loss of the CoA enzyme from the succinyl-CoA. Next, the succinate undergoes dehydrogenation to produce fumarate. Here there is a loss of two molecules of hydrogen to FAD to form FADH2. Bear in mind the FADH2 and the NADH are the electron equivalent, which are used to produce ATPs in the electron transport chain by oxidative phosphorylation. The fumarate produced can also undergo hydrolysis by an enzyme fumarase to produce malate. Then the malate undergoes oxidation once again to produce oxaloacetate. Now another molecule of acetyl-CoA comes and condenses with this compound to produce one molecule of citrate. So upon the movement of one acetyl-CoA in the Krebs cycle, there is production of 3 NADH, 1 FADH2, and 1 GTP. These are the energy equivalent, which are used to produce several molecules of ATP in the electron transport chain. So we have one glucose molecule being condensed in the glycolytic pathway. We produce a net acetyl-CoA of 2 because we have 2 pyruvates, then an equivalent NADH of 6 and 2 FADH, as well as 2 GTP in the Krebs cycle. So this cycle is the main site for the production of several molecules of the NADH. Let's see what happens here. So when you have an acetyl-CoA being condensed with oxaloacetate, there is formation of a 6-carbon molecule from acetyl-CoA and oxaloacetate, and that compound is citrate. Now, where the net or the first NADH is produced, there's loss of one carbon dioxide, that's conversion of isocitrates to alpha ketoglutarate. So here there's loss of one carbon dioxide, an oxidation reaction, and also decarboxylation reaction that leads to the production of the first molecule of ATP. Also converting from alpha ketoglutarate to succinyl-CoA, there is loss of another carbon dioxide or one carbon atom and production of one NADH. From succinyl-CoA to succinate, there is one produced GTP. Then from succinate to fumarate, there's production of one FADH2. And from malate to oxaloacetate, once again, there is production of one NADH2. So several molecules of NADH and FADH2 are produced in the Krebs cycle when you have one molecule of or one glucose molecule being condensed in the body. So one glucose will yield an equivalent NADH of 6, one FADH2, and one GTP. Therefore, let's say you have 5 acetyl-CoAs, or let's say 4 acetyl-CoAs from 2 glucose molecule. We are going to have an equivalent molecule of 4 times that is 4 times 3 here, which is 12, 4 times 1 here, which is 4, and 4 times 1 here, which is 4. So if we have 2 acetyl-CoA molecules or 4 acetyl-CoA molecules in the Krebs cycle, we produce an equivalent NADH of 12, 4 FADH2, and 4 GTP. Bear in mind, 2 acetyl-CoAs corresponds to 1 glucose molecule, 4 acetyl-CoAs correspond to 2 glucose molecules, and let's say 10 acetyl-CoA molecules corresponds to 5 glucose molecules. Thanks for watching. Don't forget to like and subscribe. In the next video, we're going to discuss how the NADH and the FADH2 are used in the production of ATP in the electron transport chain system. We're going to look at the inside and the mechanism of the electron transport system in the inner mitochondrial membrane.